So good morning, everybody. Today I'll be going over horizontal gene transfer and the role it plays in evolution. Horizontal gene transfer is seen as a crucial mechanism in driving bacterial and viral evolution, and that has led to a broad range of novel traits, ranging from adaptation to a new environment to antibiotic resistance. And most well-researched cases of horizontal transfer involves viruses, bacteria, and funguses. But horizontal transfer is particularly not well understood in multicellular animals, and neither is the evolutionary consequences between the lineages, especially because of the interactions that these novel genes are going to have across genetic architectures because it's difficult for genes to transfer into things that have a well-differentiated germline. So, and in this case in particular, it's interesting because the vector for horizontal transfer might have been a completely unrelated eukaryote. So, with horizontal transfer in animal species, it's more likely that they will be successful if we have mobile, self-regulating multiple gene copy compared to a single copy. So that's why transposable elements are something that is a good place to start exploring these horizontal transfers in eukaryotes, because in particular, P transposable elements are these 2.9 kilobase genes that are flanked by a short sequence that within itself is able to transpose within the genome and replicate multiple times over. So once it has the machinery to excise itself and integrate throughout the genome, it is easy to measure, considering the fact that with P elements, we have a unique measurable phenotype. The phenotype in particular with P elements is that we see this occurrence of hybrid dysgenesis. So although 10 to 20% of the genome of Drosophila melanogaster consists of transposable elements, a full P element in particular stands out because of its drastic phenotypic consequences, which include hybrid dysgenesis, increased pupil mortality, chromosomal aberrations, and including the usual harmful insertion mutations and decreased fitness. So with hybrid dysgenesis, what was found was in the 1960s, there was these wild type males that were caught that whenever they tried to breed them to the lab stock older females, that they were getting these dysfunctional F1s that were sterile and could produce no offspring. Yet, whenever they did the reciprocal cross of the wild stock females, and these are the P cytotypes because of this P element, versus the male lab stock, we saw normal flies. And eventually it was found that there was these P elements that were responsible for this interaction, and provided that it was a P-type female, there was a cytoplasmic interaction with the P element that was mated from the male that would result in suppression of the phenotype and resulted in normal offspring. The interesting thing about this is that within a span of essentially 30 to 40 years, that it was able to dominate this worldwide range of Drosophila melanogaster, even though there was these drastic consequences. And this is essentially down to the fact that it was able to regulate itself and its own transposition. So the good thing about P elements is that it provides an opportunity to see observable evolution in a human timescale. So it started off in the 1960s with melanogaster. By 1974, there were no M-type, so the non-P element melanogaster left in the wild. And then in 2004, the study by Bittencourt showed that although in Drosophila simulans, P elements were very rare in 2004, they became quite common by 2014. So it was a very recent invasion of Drosophila simulans, which also showed this hybrid dysgenesis phenotype. And it stands out because it was able to expand what seems like more rapidly than what melanogaster did. It became common within 10 years, while it took melanogaster about 30 years to reach that same level. And then a on top of that, they found that newer strains, more recent strains, are actually somewhat resistant to this hybrid dysgenesis, just like melanogaster are. So what I did was I expanded the search into the yakuba subclade in Drosophila, and it's a longitudinal study I did over 4 different timescales and 5 locations. So right here we can see San Tome in Bioko is where I had most of my time frames of like consistent, reliable numbers to make the analysis, but also have some Cameroon and a couple from the continental Africa. So yakuba in particular is widespread throughout the entire continent. San Tome is restricted to Sao Tome, the little island you see just south of Principe. And TSARE is found across the continent, but only in high altitudes. And these 3 species can all hybridize in the lab and in the field. And they create stable hybrid zones. And they're stable hybrid zones between yakuba and San Tome, as well as between yakuba and TSARE. So besides just surveying whether there is a presence or an absence of these P elements, the system allows us a good opportunity to see how P elements can actually transfer through between different species, between different systems due to hybridization. So in order to detect them, I had 284 Drosophila yakuba lines across 4 different time points and different locations. Then I had 110 Drosophila San Tome lines and 27 Drosophila TSARE lines. The San Tome and TSARE lines were collected in 2009 and 2013, while the yakuba lines were ranging from 2001, 2005, 2009, and then 2015 as well. And what I did was I amplified each of the 4 individual segments of this P element, because in order for it to transpose itself within the genome, you need all 4 segments to in continuously behind each other in order for it to function as its own replicator, essentially. And if I found all 4 elements, that's when I did the primers for the full gene to make sure it was a continuous segment. So what I found was that there was P elements found specifically in Drosophila yakuba, only from San Tome. So in the blue line, you can see my collections, and I started with 2005 because even in 2001 there was no elements anywhere. So from 2005 reaching to 2013, when it was collected from Bioko, you see that there is no increase at all in the frequency of, and this is a percentage at 25%. See, there's no increase at all in the presence of P elements. But when I broke it down by just location in San Tome, the island, I saw that between 2009 and then this is a little bit past 2013, so it was a most recent collection last year, 2015. I saw about 20% occurrence of a P element within the species, and it was 114 total lines that I tested from that particular location. And it was 9 full P elements, while 10 of those were partial, and no P elements were found in Bioko, and none were found in San Tome or TSARE. So ultimately, that yakuba has acquired the P element, and this allows us a good opportunity to, with like the substantial sequence information we have from the species, to study the regions of interest where these P elements are inserting. Where they inserted the P elements not known, because there's no essential genetic bridge between, let's see here, between the occurrence of between yakuba and San Tome, those can breed, but there's no functional F1s with melanogaster. And when they originally were shown to be from Willowstonii, it's thought that the sequence from Willowstonii, the P element itself, jumped to melanogaster through another vector, because they're far too removed to be able to breed as well. So the vector in particular that has been favored to explain this is this fly mite, which, because of its ecological range and the fact that it feeds essentially like as a micro syringe, is able to, within the larval pH to feed across multiple species, and that helps the P element integrate between the germlines. So what I want to ask ultimately is are all flies within each species susceptible? So we see some pretty big gaps in the phylogeny where no P elements have occurred. Did the expansion of P elements into simulants and yakuba occur through vertical or horizontal transmission? I sequenced the yakuba exons and found that the same one nucleotide that was different between Willistoni and melanogaster, the melanogaster sequence overlapped exactly with the yakuba line. And then does it preferentially insert to the same regions, or are certain regions more likely to help it spread? And then across the different species, is this change in the worldwide colonization of the P element, is it species dependent or lineage dependent? And I have crosses developed to address it from like a lineage perspective and a population perspective. And I'd like to thank everybody in the lab, and obviously thank you all for coming out this early.
Is there any questions?
Yeah, so there's no evidence in Santa Maria or TSC area?
No, I didn't find any, and the most recent from TSC area and Santa Maria I had were from 2013. But even in, like, we didn't have any from those for Santa May, because the Santa May ones were 2015. But because it was like 115 that I had from that individual issue, I was like by that point, if it's at 20%, I think I would have detected it.
When you try to find that, it's only by PCR? Or
Yeah, so I surveyed like everybody with PCR, each individual exon, and then I verified like the main full ones through sequencing. And I'm currently right now, like by the time I get back, I'll have the F1s to test the hybrid dysgenesis if the same phenotype is seen within yakuba.
how do you know this primers are somehow binding to the P element? Like if you have mutation in the primer and the P element is there with the primers and the lineage again, how do you distinguish them? How do you assure the P element is okay?
Because once the primers, so you're saying like, what if the primers don't attach?
Yes, have mutations in the P element and then the primer.
Yeah, so because I have such high numbers, and I redid all the ones from the Santa May ones to verify that it wasn't just like that one little batch happened wrong. But I did the master mixes of like a whole plate all at once, and I redo them to make sure that if I get a positive read, I redo those. And with the controls, that I'll be able to tell if the primers did or did not attach. But it's more from like a master mix perspective. Yeah, and then once it does show up, I verify just through sequencing.